Namaste, I am JV'N, Dr. ANUPAMA GOYAL. I am working as professor in Jyoti Vidyapeeth Women's University, Jaipur, and today I am delivering my video lecture on characteristics of fungi, and this video lecture is beneficial for the students studying in BSc as well as in MSc of different at different universities. Now I begin my lecture, characteristics of the fungi. Well, first, the fungi are cosmopolitan in distribution and can occur in any habitat where the life is possible. Some fungi occur in fresh or marine water, and the other fungi are terrestrial, and other fungi are airborne. Many species are parasitic, infecting the plants, animals, and human beings. The plant body of fungi consists of branched and filamentous hyphae which form a net-like structure called as mycelium, and the hyphae are aseptate and cenocytic in the class Phycomycetes, or the hyphae are septate. Hyphae are septate and uninucleate, binucleate, or multinucleate in the class Ascomycetes, Basidiomycetes, and Deuteromycetes. What I mean to say is the hyphae are septate and uninucleate in the class Ascomycetes, and the hyphae are binucleate in the class Basidiomycetes. Now, general characteristics of the fungi, we are talking about the septa have simple pores. The septa have simple pores, but in the class Basidiomycetes, septa with dolipore occur. Septa with dolipore occur. On both the sides of the dolipore, a double membranous structure, septal pore cap or parenthosome occurs. With the exception of slime molds, the protoplasm remains surrounded by a distinct cell wall. The main component of the cell wall is the fungal cellulose, also known as chitin. In some lower fungi, for example, in the members of Oomycetes, the cell wall is composed of cellulose and glucan. So the hyphae contain colorless and vacuolated cytoplasm besides endoplasmic reticulum, mitochondria, and Golgi bodies. Many non-living substances like reserve food also occur in the cytoplasm. Now, next character is fungi are entirely devoid of chlorophyll, means the chlorophyll is absent or is lacking in fungi, but carotenoids are present. The hyphae are homokaryotic or heterokaryotic, haploid, dikaryotic, or diploid. The higher fungi in the class Ascomycetes, Basidiomycetes, and Deuteromycetes are immotile. That is, the motile cells do not occur in their life cycle at any stage, but in the lower fungi belonging to the class Phycomycetes, the reproductive cells, gametes, and asexual zoospores are motile. They are uniflagellate or biflagellate. And now the flagella are of two types. First, the whiplash flagella, and second is the tinsel flagella. So students, what are the whiplash flagella? Now the question arises. So whiplash flagella, they are also called as acronematic flagella, and these flagella are a smooth flagella with 9+2 organization, And tinsel flagella are also called as pentonymatic flagella. They have numerous minute hair-like mastigonids on their surface. So in majority of the fungi, the hyphae form a felt-like structure, but in some advanced fungi, they may undergo certain modifications in response to functional needs. Some common modifications of the hyphae are: first, prosenchyma. So what is prosenchyma? The component hyphae lie more or less parallel to one another and they unite to form a loosely interwoven structure, but the individuality of the hyphae is not lost. So in this diagram you can see this AB diagram shows the diagram of the rhizomorph, CD shows the diagram of sclerotia, and E diagram shows the aprisorium. These are the various modifications which I am talking about. So I talk about now pseudoparenchyma. So what is pseudoparenchyma? The hyphae are closely intertwined and form a tissue consisting of hollow tubes running in all directions. The hyphae lose their individuality and are not distinguishable from each other. And now I talk about the rhizomorph. It is a thick strand or root-like aggregation of somatic hyphae. In this diagram, A to B, you can see the rhizomorph is there and it is root-like. So what I am telling, rhizomorph is a root-like aggregation of somatic hyphae. The hyphae lose their individuality and the whole mass behaves as an organized unit. Rhizomorph has a higher infection capacity than the individual hyphae. Now I talk about the sclerotium. It is a compact globose structure formed by the aggregation and adhesion of hyphae. It may survive for long periods, sometimes for several years, and represents the resting stage of the fungus. The sclerotia accumulate reserve food material and they help in vegetative propagation of the fungus. So sclerotia is formed by the, see in this diagram you can see the sclerotia. In this diagram C to D you can see the sclerotia, and these sclerotia are formed by the aggregation and adhesion of hyphae. And now I talk about the aprisorium, one of the modification of the fungal hyphae. So what is aprisorium? It is a terminal, simple or lobed swollen structure of germ tubes or infecting hyphae found in many parasitic fungi such as rust and powdery mildews. It adheres to the surface of the host and helps in the penetration of infectious hyphae. Now what are haustoria? Haustoria are intracellular absorbing structures of obligate parasitic fungi, and they help in the absorption of food material from the host. They vary in shape and the haustoria may be knob-like or they may be elongated, finger-like or branched. Haustoria secrete some specific enzymes which hydrolyze the proteins and carbohydrates of the host cell. Now what are the hyphal traps? The predaceous fungi develop sticky hyphae or network of fungal loops known as hyphal traps. They help in capturing the nematode nematodes. So these are the various modifications of the fungal hyphae. Now the next character is fungi are heterotrophic. So what do we understand by the term heterotrophic? Heterotrophic means the fungi do not manufacture their own food material. They are grouped into the following three categories on the basis of their modes of nutrition. So students, the fungi are divided into the three categories on the basis of their modes of nutrition. First of all, the parasitic fungi known as parasites. So what are parasites? Those fungi which obtain their nutrition from other living plants or animals are called as parasites. Some of these fungi which survive only on living protoplasm are known as obligate parasites, and the best examples of the obligate parasitic fungi are Ravenalia and Puccinia. Some other heterotrophic fungi, for example, Astrilago, Sphaerotheca and Tolyposporium, grow on the dead organic matter in the absence of living host. They are known as facultative saprophytes. Now I talk about the modes of nutrition in fungi. Saprophytes. So what are saprophytes? These fungi obtain their nutrition from their dead, decaying organic matter. Some saprophytes, such as Mucido, can obtain their nutrition only from the dead organic material, and they do not have the capacity to infect living plants or animals. They are called as obligate saprophytes. Now the saprophytic fungi, for example, Fusarium and Pythium are the saprophytic fungi, and these saprophytic fungi which have the capacity to infect living organisms, and those saprophytic fungi which can infect the living organisms are called as facultative parasites. Now the symbionts. So what are the symbionts? Some fungi grow on other living organisms and both are mutually benefited. Such an association is called as symbiosis. Lichens and mycorrhiza are the common examples of symbionts. And now I talk about this. We have already talked, and now next character is, in most of the fungi, only a part of the vegetative mycelium forms the reproductive unit and the rest remains vegetative. Such fungi are called as eucarpic. In unicellular fungi, whole vegetative cell is transformed into a reproductive unit, so they are called as holocarpic. What do you understand by the term holocarpic? So students, you say when the whole vegetative cell is transformed into a reproductive unit, it is called as, these are called as holocarpic fungi. Now fungi reproduce by vegetative, asexual and sexual means. The vegetative reproduction in fungi takes place by means of fragmentation. One of the best example is Rhizopus, Mucor, Aspergillus, Alternaria, and the fungi also reproduces by means of fission. Means the yeast. In yeast we can see the fission, and budding is also found in yeast and Astrilago. About 20% of the fungi propagate only by asexual means. Asexual reproduction takes place during favorable conditions by the formation of conidia or spores. The spores are unicellular in the fungi, Aspergillus and Penicillium, and the spores are multicellular in the fungi, Alternaria and Cercospora. They may be endogenous when developed in Pycnidia or Sporangia, for example, Mucor rhizopus, or exogenous when developed on sporophores or conidiophores. So, in this diagram, we can see how the asexual reproduction takes place by conidia formation. So, A diagram is the diagram of the Penicillium fungus, and this shows the formation of conidiophore as well as conidia. So, you can see here a number of conidia, and the B diagram is of the fungus Aspergillus. In this also it has been seen the conidiophore is there, and above the conidiophore there are bottle shaped structures called as stigmata bearing the conidia. So, thank you for hearing my lecture with patience, and I will continue in the next class. Thank you.